Thank you for joining us for this session on innovation in clinical trials. Now, if you haven't already, before you watch this presentation, I'd recommend you first watch Dr. Shannon Weston's Clinical Trials 101 presentation. It will give you useful background information for this session. Thank you to TCR2 Therapeutics for sponsoring this session. Now, today you'll join a panel of renowned experts in clinical care and research for an illuminating conversation about the future of clinical trials and why patient involvement is more important than ever. We have a world-class panel of experts, including Dr. Ron Alvarez from Vanderbilt University, Dr. Tom Herzog from the University of Cincinnati Cancer Center, and Dr. Kunle Odunsi from the University of Chicago. Gentlemen, thank you so much for being here, and I will turn it over to you.
Hey, I'm Ronnie Alvarez. I'm the Department of Obstetrics and Gynecology chair at Vanderbilt University Medical Center. And I'm here today on behalf of OCRA to present a small panel discussion on innovative clinical trials. And I'm going to let my two colleagues introduce themselves. Kunle?
Yes, thank you, Ron. I am Kunle Odunsi. I'm the director of the University of Chicago Medicine Comprehensive Cancer Center. I also serve as Dean for Oncology for the Biological Sciences Division at the University of Chicago. Thank you for having me here.
Tom?
Ronnie, yeah, thank you for having me. I'm Tom Herzog. I'm the Deputy Director of the University of Cincinnati Cancer Center and have a lot of involvement with clinical trials. One of the things I oversee is the clinical trials. I'm also chair of Protocol Review Monitoring Committee and chair of DSMB, Data Safety Monitoring Board. And also have roles in the GOG Partners and GOG Foundation as former treasurer, but now president-elect for GOG Foundation and associate director for GOG Partners, where we've done quite a bit in terms of innovative clinical trials. So excited to get into it with you. This will be fun and great to see you, Dr. Odunsi. You're amazing, and Ronnie, what you've done at Vanderbilt is amazing. So this should be a lot of fun to get some different perspectives.
Well, great to be here with both of you guys, and congratulations, Tom, as being the president-elect of GOG Foundation. You'll do a fantastic job. So today we really want to spend some time talking about innovations in clinical trials, and we picked several topics that I thought that would have us discuss in sort of a roundtable format. So one of the first things we wanted to chat about was reducing barriers to participation in clinical trials, mainly as it pertains to sort of clinical trial design. So, Tom, I know that the NRG Oncology has been spending a lot of time trying to look at eligibility criteria and testing. Do you want to comment a little bit about how that may help reduce some of the barriers to participation in clinical trials?
Yeah, it's testing for what, Ronnie? Sorry.
Like eligibility criteria, eligibility testing and exclusion criteria. I don't know if you're familiar with some of the work that they're doing there.
Yeah, I mean, I think that, it really starts with that, right? We tend to eliminate patients from clinical trials where, in retrospect, we wish we had included them. And that's where the whole movement with real world evidence is really taken on life of its own, because we get done with these trials. And one of the criticisms may be that it's a very select population, and it could be selected for patients who have a better than average performance status, for example, than patients that would normally enroll or that normally would be treated with that medication after the medication is approved. And so we have really strict testing for different lab parameters, for example. We've tended to open up the prior cancers if they don't seem to be part of the disease. However, I've heard a couple examples in the last month where people were excluded for things that were clearly not going to be an issue related to the treatment under study, and that they didn't have any active disease from the former cancer, and yet they were excluded. So we still put very long timelines on that, with even very good prognosis cancer, which is probably not necessary when there's no active disease present on exam. The other thing is that we've I'm sure we'll get into this more, but we've not had the diversity in the clinical trials that is so necessary, so that when we roll this out to the entire population, we know in terms of efficacy and toxicity, it's going to perform the same way. And so we may be missing a lot of signals by not being more inclusive in our trials. And I mean, I think that that's a huge problem in terms of our inclusion exclusion criteria that we see.
I really think that your point about it's almost like it's 1,000 first dates with every clinical trial, right? Because each of them have their unique inclusion and exclusion criteria.
Right.
And I think what the NCI as well as the cooperative groups are really trying to do their best to harmonize the inclusion and exclusion criteria across clinical trials, so that we can open it up to a larger group of patients to participate. And sometimes it doesn't even make sense. Some of the specific lab work that is ordered ahead of time, or the exclusion criteria, it really doesn't have much in the way of relevance. So I think these are very healthy changes that are happening to the clinical trial platform to be able to have a broader participation by patients. So you mentioned a little bit about diversity and the need to try to improve or cast a wider net for patients, particularly those in underserved populations. Kunle, you're a cancer center director. What are you guys doing in your cancer center, and what should cancer centers across the nation and frankly all who participate in clinical trials do to try to cast a wider net, increase our diversity, and increase the representation of the general population in our clinical cancer trials?
Yeah, thank you for that question. Enhancing diversity in clinical trials, I think it's very critical for us, because as Tom pointed out, it's very important to understand the impact of a drug or approach in our diverse populations of the United States. It is clear that depending on your background, it's possible that you behave differently in terms of how you metabolize a drug, how you excrete a drug, the type of side effects. So it's very important to make sure we enhance diversity in our clinical trials. I'll give you an example. One of the trials that was conducted in multiple myeloma, one of the major trials conducted in multiple myeloma, had a very small fraction of African-American population. And yet multiple myeloma affects disproportionately, both in incidence and mortality, the African-American population. And so the question is, how do you generalize the results of such a trial? And what the NCI has done over the last several years is really to encourage NCI cancer centers to be intentional about, making efforts to enhance inclusion, diversity in clinical trials. At my cancer center, for example, one of the things that I watch over, along with our senior leadership team very closely, is to make sure we engage the community. It requires building trust, educating the community about the value of clinical trials. Sometimes clinical trials could be misperceived by the public as, I've had comments such as, you're just being used as a guinea pig. That is far from the truth. Clinical trials nowadays are based on very solid science, requires a lot of approval processes. A lot of processes are in place where some of the even phase 1 clinical trials can provide very long-term benefit. We need to educate the public again, our underrepresented minority populations, to build trust so they understand that clinical trials could provide long-lasting benefit. And that's what we've done here. The other thing that we've done is to make sure we have community navigators who can actually help patients navigate the system, not just for their standard of care, but when it comes to clinical trials. So there are navigators in the community, and then there are navigators internally as well to help navigate the system with regards to clinical trials. Because sometimes the information that you are provided when you sign on to a clinical trial can be very daunting. You may need somebody to guide you and help you along that journey, the clinical trial journey. And I think cancer centers across the nation have begun to recognize this. And many cancer centers are beginning to make use very effectively of these community navigators to help underrepresented populations enroll in clinical trials.
Tom, I know you're pretty active with clinical trials as a clinical trial director at your institution. Are you guys doing anything in addition to what Kunle talked about to enhance accrual from underrepresented populations within your catchment area?
Yeah, great question. I first of all, I agree with everything that was said by Kunle. Thank you. That was really well said. I think that it's not only the right thing to do, it's now going to be mandatory. The FDA has issued for clinical trials moving forward. They came out in mid-April with the call for diversity plans to improve enrollment for participants from underrepresented populations. And so the first thing we really need to do is measure it. So one of the things that we've taken on is going back and looking at our measurements. I'll sort of talk from two hats here, Ronnie, if you don't mind. One being from the cancer center here. We've done pretty well. We serve, as the safety net hospital for 3-county area and so forth. So we do pretty well with underrepresented minorities in some ways, but we're still underrepresented, for example, with Hispanic population. So, it's everyone sort of gets caught up in looking at this from one direction when we really need to be looking at it from many different directions at the same time. And the way this is going to work, in my opinion, is not going to be one site's going to be able to solve all those problems. I think what we need to do, and we try to do that in the GOG, is we've now become more conscious of what sites provide what resources in terms of underrepresented populations. So we need to make sure when we're opening those trials that we have, for example, New Mexico or, a certain part of Texas or New York open so that we get enough Hispanic patients. We need to then look at, how do we get more Blacks, African Americans, those types of patients in. So you go to different centers that do it very well, but also do data well, and they take great care of patients. And so that's really, to me, a big part of this is to get the metrics. So we've been really pushing to get the metrics because it's hard to know, right? So one of the things that you want to do, where does that bar? The FDA, when they came out with everyone needs to have a diversity plan in clinical trials, didn't really tell you where the goalposts are. In other words, what's the goal for being, quote, diverse enough to meet the criteria to be acceptable to the FDA? And so one of the things that we're looking at is we need to understand what the incidence and prevalence, but more probably incidence of any particular cancer in a particular population. As we know, for example, with uterine serous cancer, we see that more in our underrepresented populations. So we need to make sure that we match that. Say it's, the incidence is higher in those patients, we really need to go above that bar then to make sure those patients are in. It's not going to be acceptable to say, well, there's only 12% in the community and that's fine, we'll just have a 10%. You may need to go higher in certain cancers where there's a bigger burden of disease because of all the things that have already been said about differences in perhaps some biologic processing and so forth that goes on, resulting in different toxicities that we wouldn't otherwise identify, and perhaps even different efficacies based on different mutations that are present. And we're finding with some of the targeted trials that are out there now that some of the biomarkers are expressed in different populations at different rates. So we need to be aware of that, because that's going to also influence how we do in terms of meeting those diversity plans. So it's a whole new world, but it's well past time that we address this. So I was happy to see that. It's a start, but hopefully we can start putting some detail and some granularity around these guidances as we move forward. But first, it's really about metrics, and it's about navigators, and it's about all the things that we said. And overcoming the trust. And that's one thing I'd love to hear from both of you, is how do we overcome that trust? I mean, that mistrust has been well earned by the history of clinical trials in underrepresented populations. There have been atrocities that have been committed.
Yeah, I would say I obviously live in an urban area, but surrounded by rural communities, which presents a whole different type of challenge in terms of trying to get our populations that live in rural communities, both, underrepresented minorities as well as socioeconomically disadvantaged populations. And we are, presently have sort of spoken hub type of development in our cancer center, where we're putting, our clinics and infusion therapy areas closer to where people live and work, and providing people sometimes with resources to be able to, like gas cards, to be able to make their appointments, and even we're going to have to think about how do we manage some childcare issues. Those sorts of things are going to be all kinds of innovations that we need to think about to be able to have clinical trial participants that are more representative of our broad population, for sure. Well, I want to pivot to, we've talked a little bit about how to open up the funnel, right, to get more patients participating in clinical trials. And I think Dr. Weston is going to be giving a primer on phase 1, phase 2, and phase 3 trials. But let's just turn and talk a bit about more innovative clinical trials. Tom, you want to take us off on that? Maybe talk about basket trials, adapted trials, and then we'll let Kunle add to that discussion.
Yeah, there's so many different things going on with basket trials of all different diseases coming in, umbrella trials where we look at a single biomarker across different tissue tumor types. So there's just a lot of interest now of making these more facile. And so the beauty of adaptive design is it really depends on some fairly sophisticated statistics that underlie this. And I'll leave it to my smarter colleagues, you two, to explain the statistics behind it. But the fact of it is that you have a probability of success with fewer patients that are put in that you can tell whether something's going to work or not. And so in an arm that appears to be underperforming, and everything's a risk, but with pretty good certainty, you can say after X number of patients, which may be well below what was intended to be enrolled, you could stop that arm. And in the meantime, in the last year, while all that was going on, there's another interesting agent that has come to the forefront for that particular pathway, biomarker, or tumor type. Depending on the trial design, you could potentially take that new asset, if you will, and slide it into the trial, which makes it so great in that you have the trial up and running already in institutions. The startup time, I don't think people are familiar with the fact that it can take more than a year in some cases, but almost always 6 months or more to get a trial from protocol done to getting patients onto the trial. And it takes quite a bit of regulatory work and all that goes into it, much of which is there for really good reasons for patient protection and everything else. Some of it could probably be cut. But back to the adaptive design, we could then slide an asset in that doesn't have to go through all that process. And so we have this new thing. Of course, we'd have preliminary data that it's safe in humans and all that, but we want to now test it if it does as well as the standard of care, for example. And so we could slide that in and take that one asset that wasn't going to work very well. And the beauty of that, and I think my colleagues would agree, is that you're no longer wasting our most valuable asset, which is really our patients. Money is important, but our patients are so important because they're the fuel that make clinical trials work. And if we could take clinical trial enrollment that's in single digit percentages now, and we could double that or triple that, the ability to discover would go up with the same scale. So if we're able to triple that, we would likely be able to discover things more rapidly, because frankly, we have more because of the great work of academic centers and what they've been and what's been going on in industry and independent labs and so forth, and the boon in artificial intelligence and biomarkers and molecular science and genetics. We have more pathways and potential compounds than we do the trials to be able to test them. And so that's why the patients have become even more precious than what they were in the past. So these adaptive trial designs, I think, are really the wave of the future, and that we can really speed up the science discovery, if you will.
Kunle, I mean, I and Tom, we both spent most of our careers doing clinical trials as lumpers, right? We'd lump all patients into one category, and we would, do a randomized trial. Maybe Kunle, talk a little bit more about the use of biomarkers to drive clinical trials nowadays. It certainly has expanded tremendously over the past decade.
Absolutely. So let me quickly make a point about what Tom said, which covers this whole innovative clinical trial design, the use of adaptive designs, and also what are called platform studies. I think it's one of the most important innovations because it's really going to transform our ability to quickly take drugs from discovery to approval. And I will come back to give some quick examples. So biomarker enhancement of clinical trials, I think we've seen some very interesting examples over the last few years where if you don't enrich for biomarker, you might actually miss the signal of efficacy for those drugs. One example is the use of tumor mutation burden, for example, where, as we all know, in general, we now test patients' tumor to understand the burden of mutation in a particular tumor. It turns out that the tumor mutation burden correlates very well with the potential for responsiveness to immune checkpoint inhibitors. So you can imagine if you lump everybody together, those with low tumor mutation burden, those with high tumor mutation burden, you're going to miss. So by enriching for tumor mutation burden only, you see patients who are able to benefit. So that's one example, and therefore tumor mutation burden has become approved by the FDA as an indication, high tumor mutation burden as an indication for immune checkpoint inhibitors. So whether you have uterine, ovarian, lung, brain, whatever the tumor type, if you test for tumor mutation burden, those tumors are likely to be responsive. That's a good example right there. So with regards to adaptive clinical trials in GYN, there are a number of them ongoing. There are two that I want to very quickly highlight that I am involved with. One is a program both are programs by nonprofit organizations, just to declare that there's no conflict of interest in talking about it on this platform. So the first is by the Cancer Research Institute, which is the leading nonprofit organization that supports cancer immunology and immunotherapy research since way back in the 1950s and 60s. So the one study that they are conducting right now is called IPROC study, which is immunotherapy in platinum-resistant ovarian cancer in collaboration with the Canadian Clinical Trials Group, along with some US investigators. This is a platform study where you can, just like Tom described, you can bring in multiple drugs into this trial so there's a master protocol where there's a new drug discovered, you can bring it in. So this program has opened, and the way the trial is, the study is designed, you have two cohorts, cohort A, cohort B, when you open an arm. In cohort A, if you see a signal, only when you see a signal do you go to cohort B. I'm sorry, stage 1 and stage 2. Only if you see a signal do you go to stage 2. And then you can bring in other cohorts, cohort B, C, D, E, and F. So this is really very exciting because there's a platform. So the one drug two drugs that are being tested right now is based on the principle that you want to reduce toxicity. You want the drug to act only at the tumor site and not affect other normal tissues in the body. And so this drug is programmed to only function when it gets into an acidic environment, such as the tumor microenvironment of ovarian cancer, which generally is acidic. So this drug does not work if the environment is not acidic, as in the case of the tumor. And so, so this master protocol allows testing of this drug in combination with multiple other agents. The second one, which is really very exciting, is by the Global Coalition for Adaptive Research. This is called Ovarian Cancer RX Protocol. And basically, the difference between IPROC and Ovarian Cancer RX is that Ovarian Cancer RX is a phase 2 and seamless phase 2-3 design. Essentially, you can have a design, a clinical trial design, again, a master protocol that allows you to take a compound all the way to phase 3, essentially all the way to a registration trial, the way this is designed. Very fascinating. And again, these are for patients who are either in the frontline setting, or patients who have recurrent ovarian cancers could be eligible for these trials. So these adaptive protocols, and some of the cohorts are driven by biomarkers. So again, you can enrich for patients who are likely to respond. And all of these trials usually are information rich. You are taking biopsies and blood tests so that you can understand very well who is responding and who is not responding, and potentially identify even better biomarkers of responsiveness. So I am very excited about these types of study design. I think is the way of the future, as Tom pointed out, and I completely agree with that statement.
I thank you so much for this really interesting part of the discussion. I think our audience is very familiar with the role that BRCA mutation status and HRD serves as a biomarker for the efficacy of PARP inhibitors. And we hope to see more and more biomarker-driven studies so that we can, as you both suggested, really select out those patient populations where these agents are going to work. Let me pivot just a bit to circulating tumor DNA, because that is obviously a very hot topic and being utilized in more and more clinical trials in very innovative ways. Tom, you want to just mention how it's being used in other diseases and how it might be used in ovarian cancer?
Yeah. Well, so it goes back, Ronnie, you and I have a history of interaction with the FDA, and you were incredibly helpful with that in terms of, you know, how well does OS, for example, ovarian survival work for, you know, for ovarian cancer? And, you know, we we really saw a dearth of drug development for a long time, almost 14 years, with just an accelerated approval going to full approval and only one new approval in the ovarian cancer space. With the, when we talk to people in industry who have to actually pay for these trials, in most cases, they'll tell us that they believed that they had to use OS and they had to, test against everything that was available. So there wasn't really a pathway forward. And so what we've been since then looking for are surrogate markers, right? Things that accurately, because it's also not fair to the patient or society to spend money on drugs that aren't working, to have something that reliably is of value. And so I think we can all agree that overall survival, if you're able to extend that with a decent quality of life, there's no one that would disagree with that's progress. But how do we do that without waiting the number of years and the thousands of patients it would take to preserve the OS effect with our long post-progression survival that we see? And so we're constantly striving to find if there are other markers of or surrogate endpoints that would help us, that would occur sooner, but would correlate well with survival. And so we're in the infancy, I think, of knowing the true role of circulating tumor DNA, RNA. You can look for, it's amazing what can be done now. In my opinion, it's something that we really need to study in these, but we don't know yet whether it's a great correlate of OS. It's been shown in some diseases, but we don't have as much track record, in the gynecologic cancers. And so I look forward to more data and then getting, the correlation with not only progression-free survival, but also hopefully OS as well. And it would be wonderful if this was a more objective way of determining treatment failures that correlate well with overall survival, because it could be used in a regulatory standpoint. Beyond that, it can certainly be used to point to patients that we need to increase surveillance or even consider maintenance strategies or something else if they don't have actual clinical criteria for recurrence. So I think there's a whole new world there that this technology opens up, and it's exciting. I look forward to the output that'll come.
I think it's really fascinating to see how it's being utilized to sort of measure those biomarkers, particularly in biomarker-driven trials. Kunle, you probably have a lot of experience in there with lots of immunotherapies, and you're looking at obviously not only circulating tumor DNA but other immunologic markers. You want to comment about how maybe you've been using it in some of your immunotherapy trials, if at all?
Yeah, so in gynecologic malignancies, as Tom pointed out, we still have not nailed down how best to use circulating tumor DNAs. Is it because they are not going to be of use? I don't think so. I think we just haven't figured out how best to use them. And I think that will come with time. In the meanwhile, the question is, what are the best biomarkers, for example, for predicting responsiveness to immunotherapies? We talked a little bit about tumor mutation burden a few minutes ago. There are other important biomarkers, but again, we have not tested them very well in GYN malignancies. So there's still a lot of work to be done. So for example, if you look at the tumor microenvironment, in fact, the initial work by our group and by the group of George Kukos, just simply looking at the number of T cells, immune cells within the tumor alone, potentially could predict responsiveness. We really have not defined all of, and then there are a lot of other cell populations within the tumor that we should look at. So really comprehensively assessing how can we use the cellular composition of the tumor microenvironment as a biomarker for responsiveness in immunotherapy. We need to begin to incorporate all of this in our clinical trials. We talk about it, we publish on them, if you do a PubMed, you find multiple papers about the role of these cells. Some are good, some are bad. How do we integrate all of this information and really use them as biomarkers? That is a gap in our field that I think we should address.
I think it's pretty interesting. Obviously, I think measuring circulating tumor DNA probably has some prognostic value, right? We're seeing that in other disease sites. What I really find interesting is the use of circulating tumor DNA to actually determine whether to escalate or de-escalate therapy. And those trials are ongoing in some of the other diseases. And I'm excited to see how effective that strategy may be to correlate with just what you said, Tom, with overall survival. And will it be something where we can not only have prognostic value, but also be utilized as a means to be able to continue or discontinue therapy in patients? And we know that long-term therapies, while effective, does come at some cost. Well,
A lot of cost in some cases, right?
Right, exactly.
Yeah.
Let's pivot to talk a little bit about patient-reported outcomes. Tom, why are they so important in the clinical trials that we do, particularly phase 3 trials?
Yes, I think they are wonderful tools that we have, or they're wonderful endpoints rather, that we have, I think, grown to appreciate more and more, because they provide the context of the patient's experience through the journey of treatment. And I think that that's really something that we hadn't always looked at. We've looked at the toxicity charts, we've looked at the efficacy endpoints, and we haven't paid attention to the impact, the true impact on the patient's life with being on treatment during the treatment, but also after the treatment. What are the long-term side effects that could be incurred, such as neuropathy, for example, from some of our treatments, and or GI distress that lasts long beyond when the drug's present. And so these types of things, I think, are really important. Having said that, we are plagued with a large amount of missing data, so we're getting better at it. One of the things that often happens is the patient has, for example, a progression, and the patient will then leave the trial, and we keep trying to get follow-up, patient-reported outcome data, which take a while to fill out, and they're either on to the next trial or on a new treatment, and many patients just don't want to be burdened with that, which is understandable. But it's then data that we lose, and then we get select data, and it can be even skewed. One of the things I've been impressed with in a negative sense has been the number of times where the eyeball test doesn't correspond with the patient-reported outcomes. I think, for example, in our minimally invasive versus open radical hysterectomy, if I remember, other than like 1 or 2 data points, there was absolutely no difference. And I'm talking, even a month after the procedure. It would seem, knowing the patients that have an open incision, and I'm not saying the trial was done incorrectly or anything. I wonder how precise the tools are that we have and whether we need sharper tools. And even some of the drug trials where we said, see, there's no difference. We give this extra drug versus placebo, and there's no difference, yet there is toxicity. Why are we not capturing that in the patient-reported outcomes? So in concept, I highly endorse it. It's something we need to do better in terms of collection and better understanding the context of that patient journey. But I think we have room for population science and for some of our colleagues in this area that specialize in quality of life and PROs to improve the science behind it so that we have tools that are more precise.
Probably utilizing better technology to capture some of that data too would really be helpful.
It would. And if I could just say one thing about that, one of the things we had at our cancer center retreat recently, we had the School of Engineering. They can now capture with 1 drop of sweat all the electrolyte imbalances, your blood sugar, everything else. We could get to the point. I mean, I have a family member who's wearing an Apple Watch and a Whoop at the same time. And you would think that, you know, everything that happens in that body is basically captured. My point being that I don't think we're too far off from very affordable technology that'll capture a lot of at least the physiology part of this in real time. We're still missing, though. How do you feel about it? How does it change your relationships? How does it change sexuality experience? How does that change interaction with your partner? How does that change depression? How does it change so many other things that we're not getting? But you're right. I think the technology is going to help us with a lot of the more physiologic endpoints.
If I may just jump in very quickly.
Please do.
One perspective also, so I agree with all of those points that we need to measure them. But one additional gap that I see is that we don't have long-term measurements of PROs, of patient-reported outcomes. We measure, you know, at the time of a trial, the trial ends, everybody goes home, and we don't have long-term 5 years, 10 years. I mean, some of our patients, many of our patients are surviving 5 years, 10 years, and beyond. So I really think one area of research opportunity that will help our patients is having this large longitudinal cohort studies of ovarian cancer survivors, where we can longitudinally track patient-reported outcomes. Because some outcomes may not be within the first, 24, 36 months when typically we close studies. Some outcomes may be delayed, and maybe you report them, way out. So the better we can understand what happens to our long-term survivors, and hopefully we'll continue to, the field will evolve so that we have many more long-term survivors. And so it's going to be important for us to be able to understand what those patients are facing.
I think one of the take-home points that I had with our previous interactions with the FDA, Tom, was one of their statement: it's hard to make an asymptomatic patient less asymptomatic, right? And so and I've always remembered that statement, and I think it's important for patients to consider that when they're thinking about treatments, and why these patient-reported outcome kind of studies are important to understand really what kind of symptoms people are getting, right, when that may be a change from being asymptomatic.
Right.
Let's talk. I've seen more and more real-world experience studies that are being presented, and you've mentioned this a bit earlier in our discussion, Tom. Why are they so important, and how do they explain discrepancies between, what we see in carefully controlled studies and what's happening out there in the real world?
Yeah, well, I think there's a filter that goes on with clinical trials, part of it due to that inclusion-exclusion process in which they write criteria that would exclude a number of patients who will, in fact, need to go on to these drugs because they'll have these cancers and they would be candidates to go on, and it's a great option for them. And so we're not collecting the data on the patients that are otherwise left out or excluded from the clinical trials, and that's certainly a big part of it. It also changes because many of the trials are done at centers of excellence. So they're done at places where there's very experienced, not only clinicians, but also support staff and patient navigators and everything else. And so they're able to help patients get through some of these more complicated treatments easier. And they may also have more means available for consultation. For example, some of our immune therapies that can affect every organ in the body. Some of the treatments and decision-making around that can be very complex and life-threatening if not done the correct way. And so if you get into places that don't have that kind of support system, that kind of knowledge base, you can see outcomes that are significantly different than what we're seeing in the clinical trial, both in terms of efficacy, but certainly in the terms of safety and toxicity.
Kunle, you've seen some of this data. What kind of discrepancies have you noted between our, published studies on the phase 3 trials and what the real world data is showing us?
Yeah, so sometimes the discrepancies are very surprising for all of the reasons that we've talked about, where you expect a certain even toxicity profile. But now you have patients who have some other comorbidities that were excluded from the original trial, and now they're exhibiting more toxicities. The other discrepancy that I have observed is even in terms of responses to the therapy. Again, this may reflect the the diversity of the population within the clinical trial, where if you look at it in the context of the diverse population, some of them may not respond for several reasons, as well as what was described in the original clinical trial. I'm sure many of us have experienced some of the challenges with bone marrow toxicity, especially for some populations, for some underrepresented minority populations compared with others. So the real world experience is telling us something about how we need to consider very carefully our inclusion-exclusion criteria. If there's one area where it's informing, it's really for us to, this inclusion-exclusion criteria, and also as much as possible to extend our clinical trials beyond these top-notch academic medical centers, to extend them to the community networks as well.
I think that's one of the insights I see is it may be easy to do these in some of the, like you said, well-staffed academic medical centers, but the implementation out there in the real world is sometimes very challenged. Well, look, I wanted to thank both of you guys for your time.
Ronnie, if I could just, I know we're getting long on time here. I just wanted to bring up this whole concept of decentralized trials. This is where we take the trial to the patient. And we have the technology to be able to do that, although we still need to overcome some of the monitoring issues and some of the things that you need in a built into the regulatory trial in terms of the various endpoints and that we need. But it's something I think all three of us would strongly endorse to meet that need of really getting a more diverse pool of patients that reflect the real world of what we're going to see. And the other thing I want to just say, it cuts across every socioeconomic class, race, everything else, is the financial part of it. And if we have financial toxicity, you may not see the same outcomes either for a number of reasons. And we have to pay attention to that as well.
Really important point. So I'm going to close just once again, thanking both of you for your time today and thanking OCRA for allowing us to talk on this important topic. I did want to make one closing statement about how important it is for patients to consider clinical trials, but also to become engaged on the front end. And I know that both of you in your roles as a cancer center directors and in the soon-to-be cooperative group setting have increasingly engaged our patients to be involved early on in the development of concepts and the development of clinical trials. And we hope that some of you that are listening to this will consider being a part of that. And I'm sure that your local cancer centers or our national cooperative groups would be very interested in having you participate in this process. It's really, really important, and we need your engagement way early in the development of these clinical trials, and not only and as being advocates for patients to go on the clinical trials and as we analyze the data. So I hope that you've learned something today, and I'm looking forward to that. We can get this group together again and talk about other interesting topics. So thank you, Dr. Odunsi and Dr. Herzog, and thank you, OCRA, for all the things that you do for our patients. Have a great day.